Welcome, everybody. Thank you for joining us. My name is Eric Testrode, and I am a member of the ADSA Graduate Student Division Education Committee. On behalf of our committee, we would like to welcome Dr. Robert Collier, who is from University of Arizona, here to talk to us about the interaction of climate, dairy cow biology, historical, current, and future trends, which, as we all know, is a pretty important topic as we deal with a warming climate. So, Dr. Collier, feel free to go ahead.
Thank you, Eric. I appreciate the opportunity. I want to thank the Graduate Student Division of the American Dairy Science Association for the opportunity to give this presentation. I want to get my slides advanced here. There we go. Go over the following 4 topics. First, throughout the presentation, I'm going to be talking about our progression of understanding about the relationship of the dairy cow to its environment, and how the list of people and scientists who have contributed to this, talk about the impact of environment on cattle, as well as we have learned the impact of cattle on the environment, and then finish up with current and future trends. If we go back to the very beginning, cattle were domesticated about 8,000 BC, and if we look at the pattern of radiation of the centers from which cattle emanated, we see 3 areas. One was the Fertile Crescent, where the Bos Taurus cattle originated from. Of course, the Fertile Crescent is in the area of Syria and Iraq. Then we have the Bos Indicus breeds, which came from the Indus Valley, and finally the hybrid group, which both Bos Indicus and Bos Taurus contributed to, referred to as Sangha, which originated out of the Rift Valley area of Africa. Now, as these cattle spread across the old world, there was the opportunity, of course, to find microenvironments, and which they did, and they adapted to those environments, and so some became more cold tolerant, especially the Bos Taurus cattle in northern Europe, and some more heat tolerant, especially those of Bos Indicus and Sangha breeds. But cattle really didn't come to the New World until about 500 years ago, and so there really hasn't been much time for cattle to evolve since coming to the New World. They mostly brought the characteristics they already had when they came. And if we look at when we actually started identifying breeds, cattle were, if we look at that process, we've got, oops, sorry, go back to that. You can see that early on, hair coat characteristics were a primary means of identifying both individual cows as well as breeds. And we know now that hair coat characteristics do play a role in how animals respond to their environments, especially if we compare thick-coated cattle with those that are from northern climates to those from southern climates who have very different hair coats. And so this process of development of breeds was also associated with differences in their response to the environment. And we really didn't start measuring production in cattle until around the turn of the 20th century, of 1900, when cattle first began to take records of the production of cattle. And this allowed producers then to start to realize that A, some animals were better than others, and B, there were some things about how they were managed that definitely affected their productivity. But it wasn't until about 1940 that we actually started to measure metabolism in cattle, and two prominent scientists who were involved in this process were Dr. Samuel Brody of the University of Missouri and Dr. Max Kleiber of the University of California. And both of these scientists were giants in their field. They really developed much of what our basic understanding is about metabolism of animals and the relationship between body size and surface area and metabolism. Both of these individuals wrote books. Samuel Brody, Bioenergetics and Growth, and Max Kleiber, The Fire of Life, Introduction to Animal Energetics. Both of these books are available. You go to Amazon, and I would recommend any graduate student who has an interest in this area to have these books in their library. They're really seminal in our understanding of metabolism, and a lot of the information that's obtained, that you can obtain in these books, we're still using today. So once we had an understanding of the metabolic rate of animals, then we were able to start to look at what is the impact of environment on the metabolism of animals. And we look at, that required to us to build the first controlled environmental rooms. And Brody at the University of Missouri built the very first set of rooms that remain, not the same rooms, but new rooms remain today at the University of Missouri. So the University of Missouri's had a continuous program in this area since early 1950s. And the controlled environments then allowed scientists to begin to look at heat stress indices, or what are some measurements that could be used to assess how stressed cows were in a given environment, and then to think about, well, how can we reduce a specific stress? And so if you look at some of the scientists who contributed to this were Dennis Armstrong and Frank Wiersma, who developed the temperature humidity index here at the University of Arizona. Donald Spires and John Goggin are continuing this work. Don at University of Missouri, Brody Environmental Chambers, John Goggin at the University of Brisbane, Terry Mader at the University of Nebraska, and Leroy Hahn, who was both at the University of Missouri and also the USDA Research Center at Clay Center, Nebraska. All of these people have contributed to developing heat stress indices as related to impact on animals, and then also when to be used to use abatement techniques. Now, if you look at the environmental factors involved in that impact animals, all of the factors shown here play some role. We know that the latitude an animal is located in has a direct impact on the degree of solar radiation exposure. Certainly air temperature, the photoperiod, as well as cloud cover, level of humidity, and wind speed, all impact an animal's ability to exchange energy with its environment. And so we had to measure these things as well. And since the early 1900s, we've had fairly good records of environmental parameters, and these also contribute to our ability to understand not only regional differences in levels of stress on animals, but also how much money we might have to invest in capital expenditure for a given area to achieve protection of animals. Now, recently we revised, using the environmental rooms here at the University of Arizona, we revised the temperature humidity index that was originally developed by Armstrong and Wiersma. And this is the new index. The old index had a stress threshold of 68, and our stress threshold is currently at 68. I believe I said 70. It should have been 72 for the old stress threshold. And this drop from 72 to 68 is directly related to the fact that dairy cows today produce much more milk and therefore a lot more heat, and are therefore also more sensitive to heat load than cattle from the 1960s when the original THI was developed. And so we not only are able to identify the stress threshold, but different levels of stress, from mild to moderate to moderate to severe, and then severe to lethal. And these then can be used in dairy operations to assess how much cooling is needed to protect animals against stress. And an interesting fact that was recently published by Umberto Bernabucci of the University of Tuscany, he looked at the effect of THI on death rate in Holstein cows, and it's interesting that when the maximum THI is above 80, or the minimum THI is just above 70, you see a fairly sharp increase in death rate in cattle. Now, this is related to the fact that stresses are additive, and when a cow has one stress, let's say she has a case of mastitis or metritis or maybe ketosis or milk fever, and then is subsequently also subjected to thermal stress, she is more likely to succumb to those stresses than an animal is completely healthy. But as a matter of fact, no one has ever really done a study to identify where cows are dying during periods of thermal stress. So an excellent dissertation might address this area of going to dairy operations and finding out for cattle that are dying during periods of severe thermal stress, where were they located on the dairy, what stage of the life cycle were they in, and were they healthy or sick. Now, heat abatement strategies are focused around two main areas. One is to minimize heat gain, and that is by providing shade. And if you're going to make one investment to protect animals, shade would be the first investment to make. Then you can reduce air temperature around the animals, allowing them to dissipate more heat and also gain less heat from the environment. And that can be done by evaporative cooling. You also want to maximize heat loss from animals, and we can use conduction where animals are lying on a cool surface. We can use forced convection with fans. We can wet cows and evaporate water directly from animals, or we can evaporate water in the air and reduce the air temperature. All of these, of course, require management strategies and capital investments. Here in the desert Southwest, we use evaporative cooling extensively. This is two systems. One on the left is fixed, and you'll notice that the cows are stationed below the coolers. We have a curtain here on the western side of the shade structure to prevent the sun from coming in and causing the cows to move, because cows will always move with the shadow, and you can see in the second system on the right, which rotates with the shadow because it has a photoelectric cell, so it can follow where the cows are. And it's one of the things that is a relatively new topic, but the study of the impact of environment on behavior is becoming a topic of great interest. And so there's a fair amount of work going on right now looking at what does a specific temperature do to the behavior of animals? Now, one thing I'd just like to point out before leaving this slide is you'll notice that almost all of these cows are lying down, which tells you they're relatively cool. Animals, when they become warm, will stand up because it exposes more surface area for cooling. So these animals are relatively cool, and this capital investment in cooling technology is having its desired effect. Now, the concept of homeoresis, which was developed by Dale Bauman and Bruce Curry and published in 1980, and if you've listened to the webinar that Dale gave a few weeks ago, that was, in my opinion, a key development in our understanding of the regulation of metabolism. And it carries over into the stress area, the environmental stress area, because when animals acclimate to a stress, it is essentially a homeoretic process. And I want to emphasize that by just looking, this was Bly, who was a physiologist, studied the impact of environment on animals, who defined acclimatization as the physiological changes in an organism which limit the effects of environmental change. He said it was based on intracellular biochemical and biophysical changes. It was chronic in nature, weeks or months, therefore not homeostatic. There was a hormonal link in the pathway from the central nervous system to the effector cell, and the effect was often to change or enhance the ability of an effector cell to respond to a disturbance. And Dale Bauman essentially said the same thing in describing homeoresis, but in a slightly different terminology. And basically what Dale said was that in homeoresis, the response of cells to homeostatic signals is changed, which is essentially the same as its bottom line is saying to change or enhance the ability of an effector cell to respond to a disturbance. So acclimatization is a homeoretic process, which means that metabolism is coordinated to support the dominant physiological process, in this case, which is to survive the stress. And therefore, if we look at, I'll just say we've done studies, myself and I've collaborated with others, looking at homeoretic regulators like prolactin, growth hormone, or thyroxine, and they are involved in regulating the response of animals to heat or cold stress. Now, about the same time, Bauman and Curry were publishing their work on homeoresis. We were looking at the University of Florida. I was collaborating with Bill Thatcher and Fuller Baser, Herb Head, and Charlie Wilcox, looking at heat stress carryover effects from late gestation into the next lactation. And this was really predicated on the observation. If you look at the seasonal trend in milk production and milk protein concentration shown here, you notice that milk protein in the blue curve bottoms out here at the hottest part of the summer. So it really represents the direct effect of heat stress on milk protein synthesis, and then it comes back up as weather cools. The red line, which is the milk yield curve, is actually composed of two parts. One is the direct effect of heat stress on milk yield, but then you'll notice that it doesn't really bottom out here until it's actually cooling off in September and October. But this is because these cows have carried out the majority of their pregnancy during hot summer months, and that adversely affects the development of the fetus, the development of the placenta, and also the ability of the mammary gland to grow in response to endocrine signals from the placenta. If we look at the seasonal pattern in birth weight, which we took from 20-year record at the University of Florida, Gainesville, and there was about 1,000 calves involved in this piece of work, and there was essentially a very pronounced seasonal decline in birth weight that bottomed out exactly when the milk yield curve was bottoming out, here in September and October. And so this carryover effect of heat stress during late gestation on milk yield and next lactation was subsequently investigated by a number of other laboratories. This is the original study shown here on the left, where we looked at a shade, no shade model, and we're able to show that just providing shade to animals during the last trimester of pregnancy resulted in an increase in milk yield. But others, David Wolfenson in Israel and several other subsequent investigators at the University of Florida, under the direction of Jeff Dahl, have shown repeatedly that cooling animals during late gestation increases postpartum milk yield. And Jeff and his group have gone on to show not only is the milk yield affected in the next lactation, but also the immune status, not only of the cow, but also the growth rate of the calf. So there's epigenetic effects involved. So this work will continue, I'm sure, for quite some time. And this group at the University of Florida has staked out a very active area of research in continuing to investigate this. Now, we also began in the 80s to think about the impact of the cow on the environment. And this really came out of the historic development of climatology in the 70s. You know, the space program really made people aware that the Earth had a finite set of resources. When the pictures of the Earth came back from the moon, it was really clear that people began to think of our planet in a different way relative to the available resources. And so the work on evaluating the metabolic rate of animals was extended to look at what does that lead to in the way of byproducts that are like greenhouse gases, manure, urine, that might affect the environment. And some of the people who did early work there kind of led the way were Don Johnson at Colorado State University, and Jude Capper and Dale Bauman at Cornell. And I just want to take a small amount of time here to talk about what is the role of dairy in greenhouse warming, because ruminants in general and dairy production and beef production in particular get some bad press relative to the impact of these industries on our environment. And some people in particular, and this is a slide I thank Dale Bauman for supplying, but it shows a couple pieces of misinformation about cattle in general. For one thing, cattle are not hindgut fermenters, so which this picture implies, but also the real impact of the dairy industry on the environment is quite small and getting smaller. If we look at how much the carbon footprint of a gallon of milk has been reduced since 1944, it's a whopping 2/3. And that's because here in the United States, we've adopted technologies that allow us to reduce the herd size, the national herd size, while still maintaining high levels of milk production. In fact, in 1944, we had about 25 million dairy cows in the United States producing milk. Today, we only have less than 9 million. So this big reduction in herd size, because we've been able to dilute maintenance costs by improving production per cow, has reduced the environmental impact of the dairy industry on our environment. But that's not true everywhere. And, you know, it's been known that productive efficiency is important for a long time. This is another slide from Dale. And this was back, you know, in 1927, the U.S. Yearbook of Agriculture, they talk about the fact that the average milk production was about 4,500 pounds per cow per year. And it said in 45 years, if they were to increase this at a rate of about 100 pounds a year for full lactation, they'd double the average production per cow and be able to provide much more milk. And so, you know, the additional supply of dairy products is shown up here needed to be produced not by more, but by better dairy cows. And that fact is still true. One other thing I wanted to point out on this slide, if you see these cows, this very bucolic scene here of cows walking up the stream, this, of course, would never be allowed today because of the point source pollution potential that might exist there. So we've changed our management practices a lot since 1927, and it's had a very beneficial effect on the environment. We've still managed to dramatically improve milk production per cow. But let me show you an alternative. I was in Pakistan earlier this year, and so I was absolutely astonished by what I saw. You know, here we have in the United States a land mass of over 3 million square miles versus a tenth of that for Pakistan. We have 318 million people in the U.S. versus 176 million people, about half of what we have here in the U.S. on a tenth of the land. We have 8.45 million dairy cattle in 23 states, producing 196 million tons of milk. There in Pakistan, they've got 38 million dairy cows and 45 million water buffalo, producing only 29 million tons of milk. They have a chronic milk shortage and a chronic shortage of forage. On the other hand, we are exporting milk and hay. So I think the lesson is relatively clear that technology applied properly with good management has tremendous opportunity for the people of Pakistan to reduce the environmental impact of their dairy and water buffalo industries, and in the process get rid of their milk shortage and their forage shortage and substantially reduce the impact of these industries on the environment in Pakistan. So a great opportunity for progress in Pakistan. Now, in the 1980s, we began to develop in vitro models to study the effect of heat stress on cellular function. And one scientist who's led the way, who developed some of the early bovine models using embryos, was Pete Hansen at the University of Florida. And he continues to publish in this area and has substantially improved our understanding of why early pregnancy, especially prior to placentation, is so sensitive to heat stress. We developed a model using bovine mammary epithelial cells, which allowed us to grow cells in culture, and they grow out in the three-dimensional ductal structures, which you can subsequently use change to a lactogenic hormone mixture from a growth mixture, and the end buds will start to develop, and they will subsequently undergo lactogenesis. You can see they swell up and fill with, you have a product here that we believe is casein that's been secreted into the lumen of these alveolar units. So you can actually get these to undergo lactogenesis in culture. And so we use this model system then to evaluate the impact of heat stress. And you can see our three-dimensional structure here on the top row in three different slides. And what 24 hours of heat stress a temperature of 42 degrees centigrade does to these same structures. You see that within 24 hours they basically collapse because the cytoskeleton has collapsed. The internal structure of these ductal trees has completely collapsed. In addition, we know through gene expression studies that these are undergoing apoptosis or cell death. So we looked at how long can these cells maintain thermal resistance, and that is defined at the cellular level by the ability to maintain heat shock protein secretion. And so if you look at the expression of heat shock protein 70, you can see that it's elevated at 2 hours, but it's starting to come down by 4 hours, and by 8 hours it's back to baseline. So at 8 hours they've lost the ability to maintain heat shock protein gene expression, and that means these cells are going to be undergoing apoptosis. So we chose those time periods to look at gene expression to see what was going on, and we also looked at, in other studies, at other time periods. And so we know at 2 hours that these cells are undergoing DNA repair, so that's upregulated at 2 hours. What's downregulated is transport into cells and also secretion. By 4 hours, protein repair is upregulated. Ubiquitination, heat stress proteins or heat shock proteins are being secreted, and we also see a rest of the cell cycle. Downregulation of all the stages of metabolism, transcription, glycolysis. We see branching morphogenesis, which requires the cytoskeleton is stopped, a decrease in transport, and also differentiation. And by 16 hours, these cells are undergoing apoptosis. So the bottom line was that we now had a model we could determine if we could alter that response by adding hormones to maybe upregulate heat shock protein gene expression, and we have identified some molecules that will do that. We also wanted to find out, are there genes that might be associated with survival, and so we've identified genes that are consistently upregulated by heat shock. And some of these same genes were identified in genome-wide association studies, looking at heat-resistant versus heat-intolerant dairy animals. And we also have animals or genes that are consistently downregulated. So it's pretty clear from the studies we did in vitro that some of the milk yield loss that we see during the summer is actually a direct effect of heat stress on cellular function. And some of it's indirect by reduced feed intake and changes in the endocrine environment, but certainly there's definitely a direct impact of heat stress on milk protein synthesis and secretion. Now, when we looked at energy metabolism in bovine mammary epithelial cells, we can see that amino acid metabolism was increased. So catabolism of amino acids, fatty acid metabolism was decreased. We had a decrease in fatty acid beta oxidation and also an increase in glucose uptake. Now, this actually agrees quite well with what Lance Baumgart and Rob Rhodes, who've done a series of studies over several years now, looking systemically at what's going on in the dairy animal relative to heat stress, and they find that in fact she changes her fuel source, so she does not burn fatty acids, so you don't see an increase in non-esterified fatty acids in plasma, even though the cow is in negative energy balance. You see an increase in catabolism of amino acids, and there's an increase in glucose uptake for energy. And so this cellular model is in fairly good agreement with what is going on systemically, and therefore we believe it's a relatively good model to continue to study the effects of heat stress on mammary epithelial cell metabolism. And this integration of cellular and systemic metabolism, of course, is part of the homeoretic process as the cow acclimates to the stress. And so we've got a lot to learn in that area, but clearly the initial studies by Lance Bongard and Rob Rhodes have opened the door to a whole new area of work in that field. Now, looking at the distribution of heat loss in cattle, you can see that as temperature rises, environmental temperature, the percentage of heat that's lost from non-evaporative means gets smaller and smaller, and the primary route of heat loss becomes evaporation. And this is because evaporation works on a vapor pressure gradient, whereas sensible heat loss like conduction, convection, and radiation require a thermal gradient, and when the environment reaches body temperature, there's no gradient left. So evaporation is the only real route of heat loss. So sweating becomes an important issue in dairy cows. We know they're not very good at sweating, and you can see that when the surface temperature here gets to 35 degrees centigrade, huge variability across the animals in sweating rate. And this low sweating rate, if you compare this to a horse can lose about 2 liters per square meter per hour, and you see these cattle only at about 150 to 200 grams per square meter per hour, so much lower than horses. So if we could increase sweating rate in cattle, we would dramatically improve their thermal tolerance and reduce their need to pant to cool themselves, which dramatically decreases feed intake. So we initiated a series of studies on looking at the apocrine sweat gland in the cow. It had never been isolated and cultured, and so we developed techniques to isolate it. I might further add, the mammary gland is a modified sweat gland, and so the structure is amazingly similar. There's one sweat gland for every hair follicle, and the other thing that's important to remember is that the sweat gland and the hair follicle work as a unit. So the structure of the hair follicle has a big impact on how much water gets evaporated once it reaches the surface. So in other words, a really rough hair coat will adversely affect evaporative heat loss, but a very fine hair allows air to get down to the skin surface and evaporate the water. So we have to look at the sweat gland, hair fiber, and a gland itself as a functional unit. Now, we've stained the epithelial and myoepithelial cells in these bovine sweat glands. You can see here that the red is staining beta-actin and smooth muscle of the myoepithelial cells, which are contractile. And then the blue is labeling the epithelial cells, which are a single layer of cuboidal cells that are surrounded by this muscular layer of myoepithelial cells. So in order for sweat to get expelled, this myoepithelial cell layer has to contract and expel the sweat. So we really don't understand what regulates either the contraction of the sweat glands or the secretion rate. So a lot of things that need to be learned. And we know that in studies that we've conducted in conjunction with several other scientists, that we can remove the hair coat and double evaporative heat loss. We can also, by controlling the number of hair follicles genetically, or potentially the activity of the individual glands, we can influence sweating rate. And then we can increase evaporative heat loss by increasing airflow over the skin surface. So each one of these has opportunities to improve heat loss. Here's a study that we did in Hawaii where you can see there's black hair coat or white hair coat, either shaved or unshaved. This is in full sun, and the two colors here are different wind speeds, 0 or 1 meter per second. So you can see the big impact of wind speed and a big impact of shaving. So this just demonstrates that the hair coat does have a big impact on the ability of animals to dissipate heat load from the skin surface. So we've developed a method for isolating bovine sweat glands, shown here. And once they're isolated, we can do receptor expression studies to determine what, this is a serotonin receptor subtypes, looking at a PCR of which receptors are being expressed in either sweat glands or whole skin, or sweat glands that came from culture. And these are the different serotonin receptor subtypes. And so we intend to look at a wide variety of molecules to see which of those have receptors in apocrine sweat glands. And we've also developed cell culture techniques, shown here. We're growing a monolayer off this sweat gland. And then eventually we're going to use what's called a transepithelial electrical resistance model to look at the impact of hormones on the tight junctions of these monolayers, because those are required in order for the sweat gland to function properly and to secrete sweat. And so getting that then will allow us to determine what regulates sweat gland function in the dairy cow. And then we have opportunities to improve, using new genetic tools, the ability of dairy cows to sweat. Here's an example. This cow in the middle, right here, has carrying the gene that's called the slick gene. You notice that the hair coat here is much different than the hair coat of her herd mate next to her. You'll notice you can see the pigmentation in the white areas of the hair coat of the skin. So the hair is so thin that you can see, and that's also true here on the tail. And the udder looks like it's clipped, but it has not been clipped. And so this animal here that carries the slick gene is about a 1 degree centigrade cooler in the warm environment of southern Florida. And so these animals were first identified by scientists at University of Florida, and they have subsequently, working with Tad Sonstergaard and others at USDA, they have identified that this slick coat gene is actually the gene for the prolactin receptor, which has a mutation. So this single mutation in the prolactin receptor gene of the apocrine sweat gland has a very dramatic impact on the ability of these animals to dissipate a heat load, as an example of how we might have been able to genetically manipulate animals to have a hair coat and sweat gland activity that would really increase their resistance to heat stress. So here's things that are going on right now that are going to continue to have big impacts. The development of the bovine genome in 2009, the genome-wide association studies that look at the genotype by environment interactions, studies in epigenetics, metabolomics, behavior or ethology. There are passive forms of cooling that are being developed that will lower the cost of cooling animals, and then gene insertion and cloning. All of these are going to contribute to improving the ability of us to manage cows in the heat. So in the future, I believe we're going to see a combination of cooling strategies and genomics. So here's an example where we're using conductive cooling. We're providing shade. We've got fans or misters, but we also have an animal here that's genetically different and more resistant to thermal stress, and at the same time would have the ability to have high levels of milk production. So I believe, in my opinion, this will be our strategy going forward, to not use a single methodology or strategy, but combined strategies to improve productivity of animals in hot environments. This is just a slide of our part of our research complex here, and also the environmental rooms. I believe environmental rooms are going to continue to have a solid future because we have a lot of work to do yet to determine the effect of specific environments on gene expression, and I think these rooms provide the opportunity to do that in a controlled way that you'll never be able to do under natural environmental conditions. So I want to thank you for allowing me to share my thoughts, and I'm open to any questions.
All right. Thank you, Dr. Collier. It's interesting to me what you mentioned about well, I guess they would be transgenic cows, as opposed to like trying to do crossbreeding with something from Africa that's more heat tolerant and a Holstein. What is the, I guess, do we know anything about what the energetic cost of, say, putting a more powerful promoter or enhancer region onto a gene that favorably protects a cow from heat stress or favorably upregulates sweat production? Is that a loss in yield that we're going to see from that as well?
The stress alters basal metabolic rate. So in order to deal with the stress, the animal has to take energy that's metabolizable energy that might go to production, has to go back to maintenance to address the stress. So in my opinion, when you, if you confer tolerance by adding some of these traits, you would reduce the energy cost of managing that animal in the environment, because they would, to stay cooler, there'd be a slight, you know, just running the sodium pumps to sweat has got some cost, but that's going to be lower than, much lower cost than what it would be if the animal had to dramatically shift its production.
Sure. Okay. And then I guess another thing you touched on was the inefficiency of other countries in their agricultural system.
Right.
Well, I assume, presumably, they're using a different type of cow than us, or are they? I don't know if they're using Holsteins or. So their cows are somewhat adapted. Is the best strategy for trying to help improve their agricultural system one of provide shade, fans, that sort of thing, or would we be looking at trying to utilize some of the technologies? Like, I don't know if, you know, for example, BST, I don't know how that affects heat stress, honestly, but if it had some effect or things like that.
Actually, we studied that. I didn't have time to go over it, but we used the environmental rooms at the University of Missouri when I was at Monsanto to evaluate how cows respond to heat stress when they're being treated with somatotropin. And because somatotropin is a homeoretic regulator, it actually improves the resistance of animals to thermal stress.
That's kind of how I
so
thought, maybe.
Their evaporative heat loss actually increased sufficiently to account for the increase in heat production associated with the use of somatotropin. But to get back to your question about what's the most efficient way, clearly, you know, they're feeding way too many animals in Pakistan, both dairy and water buffalo. And so getting the higher producing animals not only means improving the genetic base, but also means improving the management of the animals. And that's an educational process. It takes time. You know, we did it here in this country. We went through the exact same kind of change. You know, we had, wasn't that long ago in the 40s when we had 25 million dairy animals in this country. So, and anytime you can reduce the number of animals you have to feed to get the same amount of meat and milk, you're ahead. So because the dilution of maintenance that occurs, even though that animal, the higher producing animal is going to eat more, because you don't have to pay that extra maintenance cost, your savings are substantial.
Mm-hmm.
And that's why the cost of food in this country is only about 10 cents out of every dollar, whereas in Europe it's 20 cents, in Africa it's almost 70 cents.
Mm-hmm.
Because we have improved the efficiency of our production systems and allowed us to hold the cost of food to a minimum. In fact, the input cost, doesn't matter what sector of agriculture you look at, the input costs have actually gone down slightly over the last 50 years, whereas the output has increased 3 to 4-fold in most cases, in almost every industry.
Mm.
So dramatic increases in productivity, and there's some good economic analyses that have been done on that, which I'll be glad to provide if someone wants to email me and request that information.
All right, we have a couple questions here. First from Ron Kinziger. Do we know the percent of dairy farms or dairy cows in the U.S. today that receive some form of heat abatement as a management tool?
Well, if you describe the simplest form of heat abatement as a shade, I would say that 95+% of dairy operations in the U.S. would provide at least shade. Now, going from there, you'd have forced convection or fans, and I would say most dairy operations, not all, but over half, would have fans in some locations. Maybe it's only the parlor holding area, but they would have some kind of heat abatement in that area. And you know, I said the first investment is always shade. The second investment is going to be cooling for the holding area, because cows are crowded there, and because of that crowding, get more stressed. So that's typically, if you're going to look at where somebody using evaporative cooling or forced convection or wetting cows down as they wait to be milked, it's going to be in the milk parlor holding area.
All right. Second question here from, hopefully I'm saying it right, Laura Schuler. We measure heat stress with the THI by comparing thresholds. Actually, there exist different THI formulas to calculate THI, and with each formula we get a different threshold. Do we need one general THI formula to compare heat stress more effectively?
That's a good question. The differences in THI formulas are related to the fact that, say, in the southeast, you have a much higher humidity level than you do here in the southwest. And so when they have slightly different formulas, they tend to adjust for those differences. But so you should be using the THI for your region. And if you do that, then the THI value, if I say the stress threshold is 68, it should hold true regardless of location.
Okay. And then one more question, or two more, I guess, here. Has transition from grazing grass-fed industry to conventional confinement increased heat stress impacts significantly?
Well, I use a comparison. Australia is undergoing fairly dramatic climate change, and so the Australian climate's not becoming that dissimilar than Arizona's. So we've had several groups come over, and I've been over to Australia a few times. Basically, they've had to transition from grazing to confinement because of lack of rainfall. With no pasture out there, they have to go to confinement systems. And so the process is a gradual one. The kind of the route we're advising them to take, and they've chosen on their own to take as well, is to start with shades first in the milk parlor holding area, and then shade over the feed line. No shade structures per se yet, but that's eventually going to become a shade structure associated with the feed line, and more capital investment in cooling equipment, first in the milk parlor holding area in the parlor, and then eventually in the various milking groups, with the high production strain obviously getting the most investment. But to answer the question, is it more stressful? If you're providing environmental protection, it's going to be less stressful for the animal, even though she's at a higher level of production. Solar radiation is a tremendous source of heat stress. So when you combine shade along with potentially evaporative cooling, or in a confined, like cross-ventilated barns, we have very little environmental change over the year. It dramatically reduces stress on the animals.
All right. Looks like we have two more questions, and then I'll probably be out of time here. From Peter Lyle, Dr. Collier, can you tell us a little bit more about the future of conductive cooling as part of a holistic solution? What has early testing proven?
Okay. So the conductive cooling work we've done to date been primarily in controlled environment rooms. And we're trying to estimate how many watts, or what's the energy exchange that can be achieved. And I can tell you that what we've been able to achieve so far, which is about 30 watts per square meter, is insufficient to be the only route of cooling. So conductive cooling is going to always be used at this point in conjunction with another form of cooling. But if you're providing, let's say you're providing evaporative cooling or fans, and also combine it with conductive cooling, the cost, we believe, with the right managed system, and when I say we're still working on what does the water temperature need to be, and what kind of cooling capacity do you need for a specific size of barn, is going to take some more work. But the reason I think conductive cooling has got a real future is because it's passive. It doesn't require, the only energy that's required is to cool the water if you have to cool it. In some areas, the groundwater is cool enough that you don't even need to do that. The other side of it is, during the winter in northern climates, it's a tremendous way to warm cows. So depending on your location, it could be used for both. But I'm projecting that conductive cooling has a future. That's based on the assumption that it'll be economically viable. And right now, we still have work to do to make it that way. But it does have strong potential.
Okay, and looks like the last question here. There's some research about cooling drinking water in heat stress conditions. What do you think about that? Or
well,
there have
been mixed results on cooling drinking water. In some studies, cooling the water resulted in a decrease in water intake. In other studies, they've been able to, they reported improved body temperatures and improve performance. But I haven't seen a consistent set of studies yet that would say if you cooled drinking water to this temperature, you're going to get a production response. So I think more work needs to be done. The original set of studies that were done, because these tend to go in cycles, these different approaches. The last cycle of cooled water studies were done back in the 70s, and several of those reported decreases in water intake and not much change in body temperature.
Well, it looks like all the questions we have for now, and we're right at time also. So thank you again, Dr. Collier, for taking some time with us to discuss this, and thank you everyone for attending.